So, how can you slow or increase the rate of bacterial growth? Okay, the rate at which a bacteria grows or divides is by changing the temperature. If you keep food in a refrigerator, bacterial growth will slow down. So bacterial growth will slow down. When it is frozen, the bacterial growth stops. Okay. However, it's not killed. That's really important. So when you increase the temperature, the bacterial growth will come back. Okay. This is the reason why we cook everything at a high temperature, because that's the only thing that will kill bacteria. Okay. The main steps of growing penicillin in a fermenter. Okay. Put all your kind of like products that you need in the fermenter. You need to make sure that you keep the pH at about 6.7. Temperature needs to be kept quite constant at about body temperature. Okay, so that's why you have water coolers, to keep it cool. And you need a lot of air so that it can respire. After that, you kind of filter it out and extract the product and then wash and dry it. How will you grow any type of microorganism in a laboratory? Okay, the kind of first thing you want to think about is all sorts of aseptic techniques. So wash hands, close windows, bleach area, all that kind of stuff. Okay, then if you're going to grow bacteria on an agar plate is just basically nutrients for your bacteria to grow. So agar is normally pre-prepared for you. Say you're going to grow certain bacteria, you'd sterilise the inoculating loop before into a red hot flame before and after. Okay, that's to kill any other bacteria. Okay, another thing you might have is convection current to kill any bacteria. We call it inoculating. So you lift the lid, okay? Do not fully remove or place on the desk, okay? This prevents any other airborne bacteria contaminating. You lift it slightly, okay? You zigzag your bacteria across the agar. Remember, the agar is just there to help it grow, okay? After that, you secure it with strips for safety reasons, and it should be labelled and dated. Big thing in schools is we can only kind of incubate to about 25. We don't want to go up to 37 because that would encourage human pathogens to grow. So it's always at 25 in schools. For safety reasons, plates and equipment must be sterilised before and after use. We don't want to be growing any other bacteria, which is a big thing with aseptic technique. You might then be asked to look at growing antibiotics. So you grow a bacteria on the plate, then you have these little strips. You can see here, little strips, where you place an antibiotic on, and the larger the zone is, okay, we call it the inhibition zone, the more effective the antibiotic is, okay. You can see here A is the most effective because it would have killed the most bacteria. You're sometimes then asked to work out the area, work out the diameter first, okay, by measuring all the way across. Okay, so say the diameter was 10, you would then just half that to find the radius, would be 5, and then the area would be pi times 5 squared. Okay, and I've just explained that here, so you can look at how big a clean zone is. And I've also explained this, okay, is how you make sure that an area is sterile for aseptic